Welcome back. Let me take you through the lab. So I went to go get my blood drawn to give you guys a first-hand experience. The first tube that they are going to draw is a red top tube. That is a discard tube to get rid of any tissue factors from the original stick. This is a blue top tube that will go to coagulation to test how your platelets are functioning. The green top tube is going to go to chemistry, and that is where they test things like hormones, metabolites, so much more. The purple top tube goes to hematology, and that's typically used for CBCs, which quantifies your red cells, white cells, platelets, and how big they are. You'll see her mixing them really well to make sure all the additives are evenly distributed. We'll go to hematology first. We look at the whole blood aspect here, so these do not get spun down like in the other departments. We'll load it onto the analyzer. The analyzer will pick it up and make sure that it gets mixed really well, as you'll see here. And this is done to make sure everything is evenly distributed so it can quantify the cells properly and to measure their sizes. Once it's done sampling, it will push it out onto the belt for a automatic slide. And this is a machine that is had in typically larger hospitals with a lot of patient volume to save the tech time on making slides, because they are pretty tedious, as you'll see in the next few segments. The analyzer will give you a printout, and at the bottom will typically have flags if it needs you to look at the slide. So if you've got a differential that you need to get out stat, sometimes it's easier to just go ahead and make it manually instead of waiting for the stain maker to do it for you. So here you'll see me taking a super small drop of blood and smearing it onto a slide. The whole goal here is to make the smear as thin as possible, because you really want the red cells, white cells, and platelets spread out evenly so you can really get a good idea of how everything looks. So after I make the slide, I have to go through the Easter egg dyeing process. This is actually pretty cool. So these stains will fix the cells to the slide and dye the red cells red and the white cells purple, along with the platelets. So we have a finished product here of a really cool field of cells, and this is where we differentiate everything. This is a platelet right here. This is what's responsible for forming clots. This is a neutrophil, typically present in high numbers when fighting off bacterial infections, and a lymphocyte, which is really present with viral infections. That's a red blood cell that, kind of looks like a donut. And then this is what we would see with a sickle cell patient. Obviously, their red cells do not look like donuts, which is a healthy red blood cell. These are sickle shaped, and we wouldn't know this if we didn't look at it under the microscope. So now we're going to chemistry, and these tubes do have to get spun down. So there is plasma at the top, which has your hormones and metabolites, things like that, and the red cells are at the bottom. We don't really too much care about what's going on with the red cells in chemistry. So they will get loaded onto a belt into one of the cartridges here, and the analyzer will scan it to see what tests it needs run. I ran a complete metabolic panel on myself, and it has the results and reference ranges. This is what your doctor gets. Then we will head to urinalysis. So much information is had when we run these on a patient. So we look at things like liver function, kidney function, if the patient is diabetic, if they have a urinary tract infection. It's pretty cool, actually. This machine takes pictures and does a chemical strip to see what all the components are. This is an example of some of the pictures it takes. That's bacteria. These are white blood cells, and these are red blood cells, and this is all in your urine. After it's done running, those results will go to your doctor and they'll put you on antibiotics or whatever you need. Then we're off to coagulation, and this tube gets spun down as well. Not really interested in the red cells. Here we're looking more at your clotting factors rather than your platelets to see how likely you are to form a clot, having a stroke, or possibly even bleeding out if they're not functioning correctly. So this machine's pretty cool. We scan it in like on the other ones. You'll see me loaded in here. The ones that are blinking are done running, and the solid line means that this is a test that's going to be run. It will pull it back into the analyzer. This is the coagulation cascade. This is what you study when you are in school. It's really in depth, so good times. But this is what happens. It's kind of cool because you can actually see the guts of this machine. It will take a sample of the patient sample and mix it with reagents to form a reaction to get a clotting time. This is microbiology. So this is her taking a bodily fluid, something like urine that we're suspecting is a bacterial infection, and plating it out onto this plate that has food that the bacteria like to eat on. So after a couple of days, it will look like this. Then we can tell your doctor what bacteria it is, and they can put you on the proper antibiotic. This is where we put blood cultures. Those kind of look like Tabasco bottles, but they go in there, and that is like an incubator that incubates at the same temperature of the human body. Blood bank. So say you have a patient that just had surgery and they lost a lot of blood. Well, now they need some to replace it. So this is where blood typing comes in. You know, when you always hear like, oh, I'm A positive or I'm O neg. This is how we do it. So Paul here is showing you a very, very simplified version of how we blood type someone. This is kind of like the manual way to do it. We do have analyzers now that help us do it. The reagents that he is showing you is what we mix with the blood to see what type you are. So there's A, B, and D. D is at positive or negative. So depending on what clumps up or agglutinates, that is what you are. So when you see a clump, that means that that is what you are positive for. So here you can see that he has put the reagents on the blood to see what is going to clump. So he has to mix it together. Oh, so that person is A. Let's see if they have B. You see how that didn't clump up? There's no clumps like on the first one, so that does not mean that they have B. So currently just A. And no clumping for D. So that means that this person is A negative. So here we would issue them A negative units or whatever else would be compatible that we had on hand. This is simulating a trauma situation. So say there's been a massive car wreck and they need a jillion units now, super stat. This is Paul and Alex simulating a nurse and a tech doing a read-off of the patient information to make sure it's all correct, to ensure that the patient gets the correct blood. The blood will be loaded into that cooler and raced off to the operating room as fast as possible. After all that, it's time for some playtime. Thank you guys so much for watching. It's been a blast getting to take you through the lab. Please like and subscribe. It really helps the channel, and I can't wait to share more lab, travel, and golden retriever content with you soon.